Hello everyone, my name is Neha and my group members Taya and Brandon are present here with me today. Today we will talk about the topic of abscission, which is the biological process of the shedding of organs such as a leaf or fruit, and we'll demonstrate that right here. So this is what happens. I can rip it. Have you guys ever wondered why the leaves fall during the autumn? Well, today we will discuss this complex and highly regulated process through our explanations of the mechanisms, developmental processes, and biological significance. First, Taya will discuss the developmental processes of abscission.
Hello everyone. Okay, so the exact signal transduction pathway is unknown, but we do know that changes in the levels of certain hormones like auxin and ethylene heavily affect the abscission process. Now, before abscission even starts, what's gonna happen is nutrients from the target organ are going to be transferred back to the stem, from the petiole to the stem. And this occurs in order to prevent the loss of nutrients during this process. And a central part of the abscission process is the abscission zone, which is typically located between the petiole and the stem. So cells that comprise the separation or abscission zone are small, square-shaped cells that no longer have the ability to enlarge or become vacuolated during the normal growth process. These cells consist of 3 different cell types: type 1, type 2, and type 3 cells. Now, type 1 cells respond to the signal of auxin. Type 2 cells respond to ethylene only, and then type 3 cells respond to both ethylene and auxin signals. Additionally, separation must occur between a lignified layer of cells and a non-lignified layer of cells. Now, secession cells produce a hexagonal structure, lignin, and this lignin serves to localize the cell wall degrading enzymes. On the other hand, the non-lignified layer of cells secrete a protective cuticle on the surface of the plant in order to form the new epidermis. Once nutrients have been withdrawn from the plant organ and the proper signal has been sent, whether it be auxin or ethylene, cell wall linkages in the middle lamina between the 2 cell layers are disconnected by the pectin methyl esterase and the polygalacturonase or cellulase enzymes. And once the cell wall has been weakened, the reabsorption of water in the cells, as you can see here, and turgor pressure within the cells functions as the driving mechanical force for abscission. And voila, the process is complete.
So to start with abscission is the lagging phase. So in the lagging phase, the cell integrity still remains, so there's no weakening of the cell walls of the leaf, but there are changes in the cellular level that signal that we've started abscission. So first, auxin levels will have dropped and ethylene levels will rise. What happens now is that it signals mRNA levels to rise, which also causes protein synthesis to rise to generate more cellulase. And this cellulase will be later used to weaken the cell wall, and cause abscission. Also, for unknown reasons, the cytoplasm and the organelles at the separation site will begin to increase and the cells will grow. Okay, so after the lagging phase comes the separation phase. Now the separation phase has a layer to it called the separation layer, which is a non-lignified layer, which is missing several key structural proteins that are not degradable by enzymes. This is important because during the separation phase, as ethylene levels rise, cellulase and pectin methyl esterase are released, which degrade the pectin and cellulose layers of the cell wall, weakening its structure. Now you might think that cellulase is found in all the plant cells because they need to degrade the cell wall in order to grow and divide. But the scientists' studies have found that a certain cellulase called 9,5-cellulase, which has a different pH point than regular cellulase, is found in cells that are undergoing abscission. So this 9,5-cellulase is made specifically for abscising leaf cells. Now, once the cell wall has been properly degraded, the leaf still isn't abscised because it still has vessels connecting to it, such as xylose. The last step is a mechanical separation step. So what happens is the cells that are still connected to the leaf will begin to build up turgor pressure by collecting water, and this will increase the amount of mechanical force on the steel and eventually force the leaf to fall off, finishing the process of abscission.
Okay, so now we'll talk about the biological significance for the process of abscission. One of the most important biological significance for why the plant shed its leaf or other organs is primarily for plant defense against pathogens and other animals. The abscission of insect-damaged leaves can be important for the plant to conserve water and other nutrients. As you can tell by this picture, the insects have taken over the leaf, and the leaf is not functional for any purpose. Elevated temperatures and low soil water potential can also stimulate abscission due to the plant being under stress. So as you can see by this picture, the plants are under stress, and through this process being under stress, the plants will upsize, or the leaves will upsize. Some of the other important environmental factors that can trigger abscission can be the prolonged growth of, in the absence of light or drought. As you can see by this picture, there's limitations on the plant growth due to the fact that the plant cannot obtain as much light as it wants or expend a lot of energy for its resources. And then one more image I would like to show is one of the benefits of having abscission. Primarily, it's important for reproduction. Some male plants, like animal mercury plants, upsize as the anthers open, and when the flowers fall, their pollen is dispersed. So in this image, you can see that there's various fruits that are important in seed dispersal, which can definitely increase diversity in the plants. So in conclusion, as we learned before, abscission can lead to fruit growth, and without having abscission not taking place in the plants, resources can be strained and stress can cause the plant to potentially die. Also, the growth of new leaves would not be very efficient, so abscission is a very important process for this to occur.
Okay, so to sum abscission up, basically abscission is triggered by both environmental and internal stimuli that results in the loss of various plant organs. Additionally, although the exact signal transduction pathway is unknown, the process is known to take place in an area called the abscission zone, which is located between the petiole and the stem, where cells separate in response to changes in levels of various hormones, such as auxin and ethylene. And once the cell wall is weakened due to interactions with the pectin methyl esterase and polygalacturonase enzymes, the plant organ is finally separated from the stem as the reabsorption of water and increased tension on the steel causes the last remaining connective tissue to break, resulting in what you may know as leaf fall. Additionally, the benefits of abscission is that it helps aid in the reproductive processes and dispersal, along with protecting them from pathogens, disease, animal damage, drought, and so forth.